So we have people on the inner tube. Inner tube?
I found some, you get, if you want to make supplement.
I don't think Diet Coke and tea would be a good combination. You never know. So I'm like. Okay, may I have your attention, please? We need to start on time. We have a vast audience on the internet. I think we take both of them. Yeah. Well, actually, we have time to take pictures during the intermission. Let's hold the hands. Every now and
Can someone write me a message? Let me know. You hear me?
All right, let's bring the meeting to order. Thank you all for coming to the San Francisco Orchid Society, the best orchid society in West and East. Thank you all for coming, and look at this display of plants that our members have brought in. What a terrific display. This is wonderful. Please bring in your plants. If you've got plants, bring them in, show them off, and then brag about it. It's kind of fun. You don't have to bring in a dozen, but if you've got a dozen in bloom, bring it in. If you've got one in bloom, bring it in. Thank you all for coming.
No, I was.
Board director meeting is on. End of May.
Start video, that means you would.
No, somebody's talking online.
Somebody's talking online.
That
I have to mute. No, I had to mute myself. They could hear us talking.
Beautiful, beautiful. Disconnected.
Yeah, where do I want to?
I'm gonna fight him all night.
Get them all, good, okay, thank you. Anyway, the Board of Directors meeting is Monday. If you'd like to attend, you can attend virtually, just try to keep your mouth shut while we're in business. But if you got something you really want to say, then you can say it. But what we're looking for is the nominating committee is composed of myself and Derek at the moment. I'm looking for 2 more people to be on the nominating committee. We are looking for a president, vice president, secretary, treasurer for next year. So if you would like to be any of these officers, please let us know. And if you'd like to be on the board of directors, we're going to have 3 openings on the board of directors. So let us know about that too. The nominating committee will be seeking volunteers.
This is for 25 or 26?
25, 26. And I remember one time when I was president of Newport Harbor, I was without a newsletter editor. So I told the crew, I said, locking the doors, nobody gets out till I get somebody to be newsletter ready. Because I've been doing it myself for 3 months, and you know, that was when you had to put stamps on and mail. It was really a bear, but anyway, I got a newsletter editor, and she worked out great. It's wonderful. So don't make me lock the doors. Okay, so anyway, so we're looking for people to run the organization next year. There's not a lot to do, just a few things. Simple. Monday, so to speak. New people would be good if you're a new member. You have a board for you. It's kind of fun. Anyway, all right, we'll start out with Derek has some announcements. Let Derek do the announcements. Derek will then introduce our speaker and we'll be off and running.
Thank you.
Take it away.
Okay, if you brought snacks, don't forget, you get an extra raffle ticket for tonight. We've got a nice table for the game, so get that. Judging, as always, you can do that tonight, and then also in Sacramento on the first Wednesday of every month. And then I hope that you all know you've got the Pacific Orchid Expo coming up, and I want to see all of your beautiful faces and your names on that volunteer list. That's the end of February, beginning of March. Okay, we've got, looks like 5 new members. Do we have, is anyone here tonight that is a new member? Anyone online? Don't be scared. Well, welcome to the society if you're watching this recording. And with that, I'm going to introduce Daniel and thank him for coming up this year.
No,
Yeah, absolutely, Mary.
yeah, I haven't been this for a month, but I just wanted to announce, so as you all know, the Orchid Conservation Alliance offers orchids in the wild tours. And this year we have tours to Rhodes in March, Sikkim in April, Sichuan, China in June. We're going to the Upper Peninsula of Michigan in July, Papua New Guinea in August, and Tasmania in October, November. So if you're interested in any of those trips, I highly encourage you to check out our website, orchidconservationalliance.org. There's a whole bunch of pages and pictures and itineraries and so forth about our tours. And you do not have to be a member to come on one of our trips. It's a wonderful way to learn about orchids and the countries they come from.
And if you're doing announcements,
Yeah, let me just repeat this real quick. So just in case you can't hear online, Mary said, check out the full slate of stuff they've got going at orchidconservationalliance.org.
okay, speaking of announcements, in case you've been living under a rock. On the 18th, that's on the weekend, the Gold Coast Convivium Society is going to be, thank you. Get my brain started. Gold Coast Convivium Society is having a Convivium auction. This time it's a live auction instead of that virtual one. At San Mateo Garden Center, starting, I believe, at 1 o'clock on the 18th. Check our website. That's where you get all the nifty things that doesn't show up anyplace else. If people don't bring to the shows because they want to bring it to this auction. And I'm the auctioneer, so be nice. Secondly, I've plugged it before. This is kind of in the far end of our area, but if you know anybody live in the South Bay, the Malahini's Orchid Society meets this Friday at the Railroad Museum in Santa Clara. So we're just getting started. This is our third meeting since the shutdown over COVID, and now we have a place. So yay, we're back. We're back. And our people bring the best food. I'm just saying.
And then Mary, I think you sent an email recently, but I didn't get it put in the slide deck, so I'll send it out in the newsletter. But what was it again?
Oh, yeah, so, so many of you may remember Jeff. So many of you may remember Jeff Long. He had an incredible collection of orchids and carnivorous plants that he grew in his garage. And he has all kinds of, he passed away in 2013. His sister has all of these tanks, shelves, equipment, and pots, etc. She would love for them to go to a good home rather than the dump. So if you're interested, just contact me and I'll put you in touch with her. I'm gonna put something in the newsletter with her direct contact, but she lives in San Francisco, somewhere near the Mission. And, you know, you just need to go over there and pick out things and take them home.
Go ahead, add to that.
Yeah.
So, Jeff Long was a member here. He's actually better known for his carnivorous plants. He has these wonderful cases, and he has some appendices that pieces of those are still in cultivation. He's almost world renowned in carnivorous plants.
Oh, yeah.
His divisions are like, this one that's been passed around, like, I think it's like $10,000 or $14,000 for the division of this one. So the guy had high quality stuff. These cases were incredible. So these cases, although quite large, I think somebody could really.
You're gonna have to assemble them.
Yeah,
Well,
but they've been in the
he
since
built
the
them.
past, so.
Yeah.
He's gonna meet.
How long do we have to?
Not long, because they're moving, so.
Yeah,
Because the newsletter will go out on the 20th, so I can catch
Yeah,
up.
I think we should do an email blast just to send it out. He's in the process of moving.
I think
Let's
John,
do that.
wasn't John asked to do one?
Yeah, but he never contacted me about this.
I don't think we asked.
Once a month to,
Yeah.
usually it's at the time for meeting.
Okay, we'll do that.
But if we could do a blast, just send it out to the members. I'll put the information in a paragraph and send it.
Thank you.
Okay.
Okay, thank you. Now, I'm gonna introduce you. Okay, so Daniel, and I wanted to kind of infuse more of the scientific and the kind of quantitative aspect this year for our list of speakers. So he's our first speaker. He's gonna talk a little bit about systematics. And I think this is one thing that I really struggled with, so I hope you enjoy his talk. He's been working with orchids for 20 years, has some really cool microscopy, scanning electron microscopy, if you know what that means. But there's a really cool images that he's got in this presentation and in another presentation. And so I invite you to enjoy this talk about the basically naming schemes, how you can, you know, remember how to call an orchid or why it's written the way it is. And then, yeah, thank you for starting us off this year. Welcome to the stage. Right.
Here we go. Thank you for having me here. It's an honor to be the first one in your series here on the rigors of academic orchid studies. This is a 101 talk. So this is the basics. So if you're already enrolled in that sort of topics, there might not be a whole lot of new things. This is rather for the people who struggle. What is all this naming business about? There are these weird trees, but they make no sense. It's just lines and squiggles. So if this is you, this is basically what I'm trying to impress on you, what's going on. So that's what we're going with today. I also encourage you, if there's some burning questions during the talk, you know, there's some word, a key word, it just doesn't make sense, you didn't hear me. Please let me know, just let me kind of go over this quickly, and I'll gladly do that. All right. So, if this works, let's see, okay, do it this way. No. Okay, okay, overview. So what is the goal here? Becoming proficient in reading the systematic literature. So you're reading an article, trying to make sense out of it. And so then also eventually when you hear something, where you're able to make an informed opinion, yes, that makes sense, or no, this is a bunch of rubbish. Okay, that's the point. So what are the different topics that I want to cover? One is species names. So if you see the species names, they often have a bunch of other information with it. What does that even mean? Then also naming the species. How is that done? That might be possibly an achievable goal, maybe down in the long term for some people. I want to name an orchid species. Can we do it? How do we go about that? But then also the other way around, that's something I've been struggling with. There are too many names. How do we figure out when we need to get rid of names and how we do that? And then we're also looking a little bit more at the phylogenetics, so at the tree making aspects of systematics, of how we're doing that, how we're making trees, what is the basis for it, how we read trees, and how we figure out, does that make any sense? So first, anatomy of a species name. And I'm using one at Orothalid. Some of you may be familiar with it. Spiclenia brickamii, S. Watson, Pridgeon Chase, and so on. So what are all the elements in there? First one, Spiclenia brickamii. So that is the species name, or also often called a binomen. Bi for two, nomen name, the thing with two names, the genus and the species name. Then we have first the Spiclenia itself, that is the genus, sometimes also the generic name. And then brickamii, that is the species name. Now notice that sometimes we call the two top names together species names, sometimes also just the second part is the species name. That can occasionally be a little bit confusing. Or we can also call it the specific name, or even if you want to get really technical, it's called the species epithet. So then you know, okay, we're talking about only the second part.
Online or soon.
Okay, there I don't know what you're doing.
They're seeing it like.
What are they saying?
Last thing, full screen, they're seeing it.
Really?
So at any rate, so when we're working on these visual issues, then after the species name, there is the name S. Watson and then parentheses. Occasionally you see that. And so S. Watson, that was the person who described brickamii, introduced that particular species epithet. That's in parentheses because he introduced it not in the genus Spiclenia, but in a different genus. I know that was done in Orothalid brickamii, S. Watson, and you give even the reference for it in the Proceedings of the American Academy of Arts in 1888. And that original combination is frequently referred to as the basionym. Some of you may have heard that term. And then Pridgeon and Chase, those are the people who put that species epithet brickamii in its current generic placement under Spiclenia. So now you know everything, what's all that information that's contained in that long tail of names.
Can we get rid of the green and the red?
Should we just drag it down?
Okay, all right. So when we're talking about species, it helps to understand what is a species. Just a species concept. And so probably the best known one is the biological species concept that was introduced basically for birds. If two specimens interbreeding and they're producing fertile offspring, then these two specimens belong to one species. Frequently, however, particularly with plants, that's unobservable, or if we have dead specimens, we can't really figure that out, so that doesn't always apply. So then there's something called the morphological species concept. So the specimens, if two specimens look alike, then they are in the same species. Works great sometimes, but sometimes we also have, for instance, dioecious plants, where we have male and female flowers, that doesn't work. Or if we have, for instance, butterflies with caterpillar stages, then of course the caterpillar still is the same species as the butterfly, but they look very different to one another. Then there's also something called the phylogenetic species concept. So if a cluster of individuals form a phylogenetic unit, then that is a species. But for that, of course, we need a phylogeny to start with. cohesion species concept acknowledges that they're hybrids, they're certainly frequently occurring in plants. And then if they're sort of kind of hold together, then that forms a species. And last but not least, there's also what I call the expert species concept. And that is whatever the grading expert says is a species, that is a species. And so that's of course very subjective, but still today, quite frequently, that is how it really works. Let's see. Now try to figure out again how I move to the next slide, whether it works now with this thing. Okay, I don't know. Okay, beautiful. All right, naming species. Who can do that? Anybody. You can do that. You don't need an accreditation. And that can be a blessing and also a curse.
Yeah.
Because really everybody can do it. And so then sometimes there are some issues and then you need to figure that out. But there's no degree, you don't need to have a biology degree. And there are many examples of non-professional biologists who are excellent systematists. I want to emphasize that. And there are some people with degrees who maybe shouldn't do it. It really goes both ways. The only thing you need to do is follow the International Code of Nomenclature, ICN. So what you need to do is you need to have a specimen base. You need to have actual specimen. You can't just make it up. Okay, that's fairly reasonable. And then also, particularly with orchids, please have your specimens being legally obtained. There are some famous cases. We don't need to go there, but be aware that there are some issues there. And then also it needs to be published. You can't write it down on a plant tag. No, sorry, that's not how it works. You need to publish it, and there are certain requirements for doing that. Do you need to have it registered? No, actually not yet, at least. In zoology, that's slowly coming in botany, I don't think quite yet. So let's look at the species description and look at it, how it looks like. So this is for an encyclica. You see there, it's published in a fairly well-respected journal in Phytotaxa. We see here Fimbriata, a new large-flowered species. So it tells us right in the title, we're talking about a new species. So then the actual descriptions, let's apply all our knowledge for the terminology that we developed. We have the genus already at first. In Cyclia, a species needs to be placed in a genus. You can't just publish a species on its own without assigning to it a species. Then you have in green the species epithet, and afterwards come the authors. So you acknowledge these are the people who actually introduce it, and then you also tell it SP Nov, NSP, something of that sort, new species, you can spell it out. You need to tell the world, yes, this is an intentional introduction of a new species. And then you have a diagnosis. And in the past, until about 2010, I believe, it needed to be in Latin. Nowadays, you can get away with that. You can also do it in English. Hooray, yes. We're making slow progress, even in systematics. And then in brown, underneath in the brown box, you see type. And so that is the type specimen. So that is the specimen on which the species is based, where it was found. And in the green box, Thomas et al. 14604, that is the collection. So the collector, Thomas, collected it, and some helpers. And they have a number that is their collection, a so-called gathering. And so that is the particular specimen that is used for that. And we have also what sort of a type is it? There's a whole nomenclature about types. This is a holotype, and it tells you it is deposited in this CEPEC specimen. Exclamation point means we have seen it. All right, everything is good. And then at the very bottom, you see also the etymology. So the meaning of the species explained, doesn't need to be, but it helps to actually justify and explain what did we intend to mean with that. Yes, Matt.
So you were talking about having it published in a reputable. So who decides what is reputable?
Excellent question. It is pretty loose. So publication can also mean a book. It doesn't need to mean a journal. So you can publish in a book. There's also digital opportunities to publish it on CD-ROMs. There are some opportunities. Be very careful with that, though. So particularly with newsletters, that may not count as a publication. In general, if it's peer-reviewed, that is already a good sign. So again, kind of Orchid Society newsletter, with all due respect, of course, is not the right place to do that. So if you're not sure, possibly check with a colleague, someone who has done that before, and get some feedback. That's probably my best advice, but there's quite a bit of leeway given there. And we're getting actually to just, I think in the next, no, not quite yet, but we're getting towards about the acceptance of species in just a moment, if you just want to bear with me for another moment. So now, of course, a picture says 1,000 words. Illustrations are helpful, but they're not mandatory at all. And so and so now that's where we're getting to. Is the species actually good? Does it hold any water? So there's no pre-publication commission. That is for instance, in contrast to mineralogy, where in mineralogy, there's a commission that decides, is that mineral actually new, or have we seen that before? In the zoology, the life sciences, and in botany, that does not exist. Then, however, the peer review filter hopefully helps us to really weed down the problems with certain species descriptions. Either the species is already known, or there are some ambiguities that could be clarified in the original description. And thereafter, it's what I call the court of public opinion. Everybody in the community decides, yes, I think this is new, or no, this is rubbish. And so then, of course, many people say, okay, there's the gold standard, then of course the Q list, or now Plants of the World Online. That is one opinion, okay? And even AOS endorsement does not mean that this is the one and only gold standard. You can make your own opinion. And of course, that is headed under the keyword of freedom of taxonomic thought. You can make your own opinion. You think, nah, I don't believe that species, or that actually should be separated into three. Yes, that's your opinion. You're entitled to it. So that then brings us to, the other way around. The other way around, synonyms. And so this is basically what I'm battling with a group of flowers that has been last revised in 1859. And so globally, and so there has a lot of debris accumulated. And so one good approach is that basically on global average, there are about 3 synonyms for one correct species. So every single species has on average about 4 names. So if you have a group, say genus, where 80% of all the species are considered correct, Houston, we've got a problem. Maybe we need to look at that a little bit more carefully. Now, which one of those names do we choose? The best known one? No, it's the oldest one. We always give deference to the oldest one. There are some exceptions, that's for later. And then the question also, the question is not how many species are there, but how few. It's a very different kind of mindset. And that is often referred to as the lumper versus splitter mindset. So do you think rather there are more variable species, but rather fewer, or there are many very small divided species among there? So I want to give you a few examples here. Also thinking about particularly what is referred to either typological thinking or population thinking. Typological means is basically we have one reference specimen, the type specimen, and every ever so small deviation, that's a new species. Or we're thinking, oh, well, nature, there is natural variability. That's what Darwin required for us for evolution happened. There is variation. And so therefore, we're thinking rather about a species being composed of a number of specimens with some variation. And so then the big question is, okay, how much is there natural variation? How much is there biodiversity? So this is the struggle that we are facing when we're doing systematics, when we're trying to figure out how many species are there for real. And one carrot I show you here on the right is an inflorescence of Obronia ceticera from the Philippines. And you see at the top, the flowers in whorls, and the bottom, the flowers in spirals. And these characters, either in whorls or in spirals, have been used to as indicator for different species. And now we have it on one and the same inflorescence. Obviously, one specimen is not composed of 2 different species. That is pretty clear. So there we have some fairly good indications of interspecific variation. And that's also where actually we here as growers come in, because we are growing specimens over multiple years in our greenhouses, in our outdoor enclosures. And so we see in between year variability. And as opposed to typical, the field collectors, they see one plant, they take it, and then that's it. They don't have a repeat sampling there. And so actually the cultivation can help us with understanding this question of biodiversity versus variability. And also, we also need to remember that particular type specimen are something called the SEMA form. SEMA is the meaning that the signifier foreign to carry, so the carrying of a name, of a meaning, but not an immutable unit. So now, why might there be synonyms? And I want to give you some examples here. And one of them is not checking the literature. And if you hear Hypnothelum macranthum, we see on the right the description. And we find first introduction, the description, and then acknowledgement. Where's the literature? None. And so if you see basically no literature cited, you may think, maybe you should have done a little bit more homework. And so yes, no surprise, that species turned out to be a synonym. Second one, bad drawings. And so I show you here Katrina in the center here. This is the description that was, the drawing that was published in 1981. This is the actual specimen out here. Where, for instance, we see this very narrow horn-like petals. They're actually triangular. We see a fairly kind of shallow notch here in the epityle. This is actually very hammer-shaped. And once you see the specimen, you suddenly realize, oh my goodness, it's the same as these guys, and that's what it looks like under the scanning electron micrograph if you do vote for that route. So bad drawings is another source. Then also sometimes you just ignore other people who have a little bit of knowledge about the group. So this is the example with Oberonia chloride. This is this particular specimen. This is the type of a new species that was proposed. However, it looks the same like this one, like Griffithiana, with very nice illustrations by Lindley in 1838, widely available on the internet, live plants over here, electron micrograph on top. So that's exactly the same. Yes, ma'am.
Who's policing this?
The person who looks after it. So that was not policed during the review stage. So that was basically a failure of the peer review system. And so then basically someone like me is coming in and cleans it up afterwards. But yeah, I agree. You may wonder, how did it happen? And so anyway, but it does. And so now we're correcting that. And then another one is, same example here, a comparison, inappropriate comparison. So this species was compared to that one. And yes, it looks different. And so because these two look different, therefore it's a new species, logical, right? However, if you would compare it to this one, then you would have a harder case of making the distinction. And so many reviewers, particularly in groups that have not been monographed, for which there's not the book that you can just check it out, it takes a while to look through all these 470 names and figuring out what it is. Now, on a side note, here, this species here, Cavallierii, it is on the plant opportunity table. So if you want to grow this one and you don't have it in your collection yet, you may find that today in here coming home with you. Another one is assumption of endemism. Endemism means restricted range, and it only occurs in one particular island in this case. And so here we have all these different species, all these names that are all found, one on Sumatra, one on Java, one on Celebes, one on Papua New Guinea. And once you look at them all, you see, hey, that's all one and the same thing with much wider distribution. And there's a second case for that, that's Hypnothelum. That used to be called Hypnothelum macranthum with a bunch of synonyms. Now it's reduced to Oberonia hungarius, every single one occurring on one Indo-Pacific species island. And now we recognize that this is actually a widespread species. And this is how it looks like live with very nice long inflorescences. And if you like this one, it's also on your plant opportunity table. It may follow your own, even properly named. Yes, we like that. This way. So synonyms, so what? Few more names, stand collecting at its worst, right? So, however, I may beg to differ here. One, for instance, is just how many species out there. That's kind of an important metric on its own rate. There are also questions, for instance, in particular areas like California is a biodiversity hotspot. Is it really? If you don't have the right species count, if you don't have the right species concept, we may actually have the wrong centers of endemicity. And that may, for instance, misguide conservation efforts. If you basically assume that this particular area needs to be conserved because a lot of different names occur there, but all these different names actually are all referring to one and the same species, you're wasting your efforts. And then there's a question of, yes.
So Dave, I was in Western Australia and had the spider calodemias, and I was with the gentleman who wrote the book of the orchids of that region, and it was interesting. I thought, well, how do you tell this one from that one? And he kind of like took me aside and said, well, you know what, we do this. This is the only way the government will preserve land. They differentiate, they give them different names, but taxonomically, it's going to create a problem down the line. But preserving wise, they can set aside more land. So it's really, it's a quandary that way, because you're protecting the species by giving them different names, but yet they're the same thing.
Yeah, so for those of Zoom who may not heard that, so there's also an aspect of giving different names so that you can afford them more protection. That's a good idea. Now, I'm coming from the pure systematic perspective. I want to know what it is. I don't have an end goal. I want to know what the quote-unquote truth, if you want to use that big word, using it. So that's kind of my approach that I'm coming from, and from the really unapplied sciences. So
My question for you is, do you tend to be a lumper?
I tend to be a lumper, yes.
Yeah.
Definitely. So.
Sorry, I call you.
I've
Definitely a
also-
lumper.
I've described about 100 species, particularly in mollusks. So, and I've also described it based on few specimens, but if it looks the same, I call it the same. So a rose is a rose by any other name. So that's kind of just my bias, absolutely. And so this the example in actually with Australia is quite appropriate, because I show you here, this is Obromia attenuata. This is insectivora. You tell me the difference. Attenuata was called extinct in Queensland, has been miraculously recovered. I would call that just a peripheral isolate. So it occasionally expands into the margin of its distribution. However, it is a widespread species in the Malayan archipelago. So there where we see kind of these conflicting interests between land conservation, between giving things different names. But from my perspective, we need to know what is, and the distribution of one species is different from multiple smaller species. So, all right, leaving that part behind us, and yes, it's very contentious, I will understand that. So if you look at the different species naming disciplines, there are also some confusion. Typically systematics, that is really the overall reaching term. Anything to do with how do we put an order onto nature, that is systematics. Nomenclature, that is just about the names. How do we spell the name? What is the correct name? What is the oldest name? And that is the business of the International Code of Botanical Nomenclature. And then what's frequently confuses nomenclature and taxonomy. They're almost used interchangeable. Some people make a distinction. Taxonomy is particularly the question of what is a species, how far or how wide, how narrow do we define the species, where do we assign the species to a genus. That's also referred frequently as beta taxonomy, alpha taxonomy being at the species level, and beta taxonomy at the higher level than that. And last but not least, phylogenetics, that is then the tree making aspect that we're getting into next. Sir.
I just wanted to get a clarification on one species that I have used a lot, and that's Lelia ansa, which now I understand is no longer Lelia, and what is it?
I know Lelia has been quite controversial. I don't know the latest on that. There I would also have to go into the primary literature and see what's going on. But there basically we're looking a little bit, most likely this is based on phylogenetics. And so now we'll provide you a little bit of tools of how do you read that literature, and how do you kind of figure out for yourself, should I call my plant Lelia anceps? Is it a Cattleya anceps? I don't even remember all the different names it's been under.
Because for a while, all the Cattleyas we knew were Sophronitis. I remember for some years, right? And then they were saying all the South American ones are all Lelia. Right. And then all the Cattleyas also switched into Sophronitis for a long time.
That's why my
But that was
labels are,
so confusing.
that's
I
why
was
my
like,
labels
what
are
is
in pencil.
happening?
Yeah.
What is happening?
Right, exactly. So this is basically based on phylogenetic studies in general. And so now let's look a little bit how we can make sense out of these phylogenetic studies, so that we don't have to just take the word of one person versus another one, that we can actually come to our own conclusions. So in phylogenetics, basically what we're going to do is reconstruct the family tree of organisms. There are also some adjacent subject matters and areas of studies, such as biogeography. And so that is, for instance, the geographic relationship and the geographic evolution of different species. Then there's something that sounds very similar, phylogeography, that is the same process, but within species. And then if you go even finer grained, and so I put that in gray, that's population genetics. That's then all the genetic variation that occurs within a species. I leave that aside. We're just looking at species and above. So with phylogenetics, so what do we have as raw data? We can have morphology, gross morphology, flower morphology. We can have ultrastructure. I show you here just a placeholder, a transmission electron micrograph, but nowadays it's essentially molecules. Well, yes, there's still some morphology going on, but essentially everything is based more or less on the DNA. Genetic information, DNA, RNA, proteins, sometimes secondary metabolates, rarely, but they do exist. So with DNA, there's a lot of terminology associated with it. So let's clarify that briefly. So DNA, this is basically the molecule that makes the genetic code composed of 4 letters, A, G, and T. And then we have also DNA in different areas of the cell. I show you at the bottom a cell diagram. And depending on from which particular portion of the cell we are taking the DNA, they occasionally have different prefixes. nDNA for nuclear, MT, you may have seen that for mitochondria, the energy producing organelles, chloroplast DNA for CP, ribosomal DNA with rDNA, and then coding, non-coding. Some of you may have seen that. Coding means basically it's made into a protein, non-coding is not made into a protein. That's all there is to it. And then a gene essentially is a functional unit of the genetic code. There are also some, if you're going to say, non-functional unit that you may have heard of. One is so-called SNPs, SNP, so small single nuclear polymorphisms, and then UCE, ultra-conserved elements. These are also pieces of DNA that can be used in phylogenetic context. Yes, ma'am.
Question about the organelle.
Yes.
In mammalian cells, mitochondria kind of are slightly independent of the rest of the cell. Is the same true for chloroplasts?
Yes, to a certain degree, yes. Yeah, they can be even cultivated outside of a plant, as the case with sea slug, the solar-powered sea slug, the legiates, if you may be familiar with it, that can actually extract just the chloroplasts and store them, and then the chloroplasts make actually sugar for the sea slug. Mind-boggling, I still can't wrap my head around that. So yeah, there's a certain independence there, but it's a partial independence. And that goes then back to the symbiont theory of how it all evolved. That's for the 200 level series, okay? All right. And then we look at, of course, the genome. That is all DNA. And one of the important things that we realize nowadays, we're in the age of genomics, where basically we can extract all the genetics, all the genetic code. And so the question then occurs, okay, what then if you have all the information? That's it. And so this is kind of an interesting quandary right now, where we're at the basically at the frontier, where basically we have all the data and there's nothing else coming. So how are we moving forward? Interesting question. I have no answer for you. All right. So how is DNA data obtained? So you extract all the DNA from a plant cell. You can do that mechanically, chemically. Then you can make multiple copies of particular fragments that you're interested in, and that's the domain of something called the polymerase chain reaction. There's another way you can do it with bacteria with something called the cDNA library. And then you read these pieces. And I'll show you that in a fairly old-fashioned way. This is from the early 2000s. That's a plate sequence. If you've seen that. So nowadays that's really outdated, but at least it's a good visual. And so you see these different color bands. These are referring to individual bases, and you read the individual bases, and that gives you the sequence of your DNA. Easy. All right. Now we have DNA sequences. Then we're making it into a data matrix, and the data matrix essentially is a spreadsheet. In horizontal rows, we have the species that we're interested in. And in a column, we have the characters. It can be morphological characters, or it can be a particular DNA base. And the cells then is referred to as the character state, a particular expression of a particular morphological or molecular feature for a particular species. And so we have then the alignment issues, and we can see in here in red, we have informative characters, in blue as well with indels, and then all the white ones, these are all the same, so they're uninformative. So they're not really telling us anything about it, they just come along for the ride. And then we're making trees. And selecting trees, that's almost a philosophical argument, let's not get into that. So essentially, there are two different methods. It's, if you want to call it evolutionary methods, logistics, also occasionally referred to as maximum parsimony or parsimony, that's mainly used for morphological characters. And then two methods called maximum likelihood and Bayesian analysis. And that's basically just mathematical ways of crunching the data. There's also something called non-evolution methods. They are referred to as UPGMA or unweighted paired group method of arithmetic means, neighbor joining, and also cluster analysis. Different approaches that, however, are not taking evolution explicitly into account. Getting into the details of that, that's what I do for grad school too. So we'll not go in there. So just make that distinction: evolutionary versus non-evolutionary methods. And then how do we select the tree? We're selecting the one that assumes the fewest evolutionary changes given the data on a particular tree. And so I show you that with here a very small data matrix. You have DNA and morphological characters 0 and 1 coded for 3 species. And we see that in one tree on the left, it requires 3 steps of 3 changes to account for the observed data matrix. In the other one, we have 2 steps. So 2 steps is fewer than 3. We select the one with 2 steps. Very simple. Okay. That can get quite a bit more complicated by calculating costs. And I just want to give you a quick example here. Particularly on the molecular side, some of you may be aware there are 2 types of DNA bases. And so based with 2 rings or 1 ring, and switching between the 1 and the 2 ring is more difficult than switching just between the 2 ring or among the 2 ring versions. So we can assign different costs to that. And then we can take these sort of cost matrices to the nth degree and have much more complicated models today. That's basically what's going on today. Now let's look into how we read trees. And one of the first things is how do we express uncertainty? And that's referred to as a polytomy. So this 3-pronged stem with 1 horizontal line. And from each of these lines, the X, Y, and Z's are equally distant from one another. And that means that we have downwards, you see that sloping arrow, we can have them in 3 different configurations. We can either say that X and Y are closer together, we can either have Y and Z closer together, or we can have X and Z together. All compatible with that 3-pronged approach. Additionally, we can also look at moving these nodes. Rotation at those nodes is important to recognize. And so you need to always be aware that here X, Y close together towards Z and Y, X closer to Z is exactly the same. So the idea here is think about it called a mobile, okay? If you can rotate all the mobile around, it may look a little bit different, but the pieces are the same, they're connected in the same way, they show the same relationships. We're not moving, say, that red ball over here, then we have a different relationship, and that would be a different evolutionary relationship. So thinking about rotating this nose is very important to recognize. Then we also need to occasionally understand how uncertainty is expressed, and that is essentially done with certain statistical numbers. Typically referred to as bootstraps, there's some other ones as well. And typically you see something like 100, 100, maybe 95, 100. Yes, we like that 100% support, but sometimes you see something like a hyphen here, unsupported, maybe 65, not so good. So therefore, if you see something like this, we know actually, we should rather consider that a polytomy, and so therefore the relationships are not as well supported by the data. And then this polytomy can again be resolved in different ways, and we can rotate at every single node. All right. So now we have all, yes.
And this is, the data is also based on something with the help of, for example, like molecular.
So the data is based on molecules with clocks. We're getting into that in just a moment. Sometimes there's a clock model involved, sometimes not. That's in the 500 class. We're not getting there today. So just, yes, you're right, there are occasionally molecular clocks involved, but that takes a little bit more. So now let's read one of these papers together to see what's happening. And we're looking here at a phylogeny of Cypripedium. So slipper orchids, we like that, yes. So what did I find out about slipper groups? So you can first, what you want to do is you want to see that the outgroups, so the comparison material to the slipper orchids themselves is appropriate. And so if you look at that, we're having down here the apostosioids. Apostosioids are used, and so these are the basal or orchids, non-orchids. There's all this discussion about this, but I stepped that aside, but that looks reasonable. Basically, what you want to see is that the outgroup is not something like corn or yeast, something really far apart, because that may skew the results. Then materials and methods. There's a lot of techy details about how particular DNA is extracted. Forget about it, doesn't matter, unless you want to get into that. If you're in grad school and you want to develop the protocols, then you read that. But as an orchid grower, don't worry about it. Now, then maybe look for the methods. Okay, what sort of analytical methods were used? And we see here phylogenetic analysis was used with maximum Harseman and Bayesian. Yes, to evolution methods. Yes, we like that. Okay, good. Pass. And then we're looking at the figures. So we're looking for support values at possibly large than 95. Be aware that if it's below, say, 85, maybe 70, you may think about there are some issues. If you see different figures, why are there different figures? Because, for instance, there were different analytic techniques used. Sometimes the different sort of data used, say the mitochondrial genes versus the chloroplast genes versus the nuclear genes, sometimes all together. So that is frequently what the different figures are used for. And ideally, in the ideal world, every single result should exactly show us the same result, but that's never the case. And so then that can also give you an indication if you see, one data set, the mitochondria tell you one thing, the chloroplasts tell you something else, and okay, maybe this is not so solid. So, all right, here we go. And for you, I color coded. So we have two here, two of these trees. And one is ITS, is a particular marker, a particular gene, the internal transparent spacer. And then we have the chloroplast DNA for the other tree. So that's basically the difference between those two. At the top, you see we have Tibetianum, and that Tibetianum is actually showing on the other tree down there. Is that a problem? No, it's not. Because remember again, rotating at nodes. So then you need to follow down the Tibetium. Okay, it's coming down here to that node. Here it's at this node. And if you just flip one around, then they show the relationships. That's the same. So in a sense, this is poor editing. So as an author, as an editor, I would ask the author, please make them as compatible as possible. But you as the reader should still be able to figure this out. Okay? So think about this rotation of nodes. and of how can we make them the same. But then we have all these other groups here. They're kind of scattered all over the place. So we have this agreement in some groups. And then also last but not least, here we have our outgroup, Apostasia is here. Apostasia is not in there. What happened? Very simple. It didn't work in the lab. So there's not necessarily a big mystery why a particular group has not been used. It just, well, we couldn't get the data. It happens. That's just real life.
Daniel?
Yes.
So what's your take? I know that your learn is obviously a specialty of yours, but with other genera of orchids, what's your take on because obviously you've already said you're kind of a lumper. So how do you feel about the all the genera come up, like the split of Erie and Dendrobium and Bulbophyllum, and not only the separation, but also the dumping them back into these mega genera. What's your take on these? Do you have an opinion on these mega genera? Because they're not really convenient, they don't help people learn or understand relationship. So what's your take on that?
Okay, the question is, how do we translate a phylogeny into a classification? And there are some larger genera and some smaller ones, some smaller units, just repeating for the Zoom people. Hopefully you didn't hear that. What is my take on that? It depends. And there is not the correct way of doing it. There just isn't.
Yes,
There's a certain amount of, this is the best I can think of. There's also, think about genera, okay? There's a subgenera, so you can have smaller pieces that you can use to define them. And then frequently also there's the question, there are kind of two options. Either you have one mega genus, or if you really want to keep all the groups as so-called monophyletic entities, we're getting to that in just a moment, so wait for it for just a moment. Either you want to keep the old genera, but then you have to introduce 15 additional ones in order for everything to be reciprocally monophyletic. And so the question is, which of these bad choices do I want to have? Do I want to have one mega genus that is unwieldy, or do I want to have basically 50 genera in phylogeny, for instance, that all have two species? What is more useful? I don't know. And now with specific cases, I haven't really looked at it. I'm kind of a single-minded person right now, particularly as I'm writing my book. I'm focusing on that one thing. So I don't have a particular opinion on that. I just want to give you the tool of how can you make find your own opinion on that, of making yourself through the literature.
I think one error that comes into all of this, the specimen that you're doing sequencing on, it wasn't correctly identified in the first place. For example, that's what happened with Sophonitis. They used the type for the genus, which was Sophonitis stenuba, which is completely different than all of the other Sophonitis. But they used that because it was the type for the genus to lump everything together into Cattleya. If you look at the actual sequence data, all the rest of the Sophonitis come off as this very distinct branch of the B, still Sophonitis, except the type is not. So it either had to create a new genus or whatever, but I mean, that happens. And then there's the case with the Adonic blossoms where there was misidentification of the type that was used. That's created this whole controversy about whether Adonic blossoms should be Lancidians or not. Everybody's fighting over it. And so it's absolutely critically important that whatever you use the sequence data from, you identify it correctly in the first place. That just creates more mess.
Yeah, I couldn't agree more. So the issue was, again, repeating for the Zoom people, the importance that material is properly identified. And that, of course, also means that the material is actually vouchered. Because frequently you just see a name, and then you hope that a person actually properly identified, but you cannot possibly check it. And so that is a big issue in general. And there are some estimates of how much is improperly identified in GenBank. So that's a whole different story. I hear you absolutely loud and clear. Don't have any particular opinions on particular cases. Again, I'm giving you here the tools to read the literature yourself in an informed manner. So anyway, so here we have these two trees. If you now look at the combined data, I want to show you that as well. Of this very same study, and these certain groups are well agreed upon. And then we have also certain groups are placed due to the core class, some are placed due to the ITS sequences, and then also some places is a new one. And so this is something you can look at if you're interested, for instance, in the subgroup of the Cypripediums. Now we talked also about support values, and particularly here, common issue is that here the back these lower nodes, so also referred to as the backbone, is poorly supported. And you see here all these dashes. So essentially, all of this is basically one big polytope. So that means that although these groupings here may be well grouping together and held together, the relationships among them is poorly resolved. So now you know, okay, more work is needed. Maybe that data was not sufficiently good. This is the take-home message from that. So now getting to what Ron was talking about, classification. So once we have a tree, one of the things we agree nowadays that we should only name monophyletic groups, and that means an ancestor and all its descendants. Now that means, for instance, here D, E, and C, G, H, and F, maybe even including those together, or even going all the way to the base. But if it includes A, it needs to include everything. We cannot have just two things that have special characters but are not related to one another. Or if you have an ancestor but not all of its descendants, that is also something that we are not naming anymore these days. Now, the big question is of how deep do you go down to group things in a genus, and that is really subjective. There are no really good objective criteria for that, and that is frequently where the battles lie. Then also, once you have a tree, okay, hooray, now what? So one of the things we can do is character state reconstruction. We can also map the characters. We can use dating with molecular clocks that was alluded to, and then also work in biogeography. I just want to show you a few examples here. And this is, for instance, character mapping. In this case, when we map the character, we're using, for instance, DNA data, molecular data to construct the tree. And then we are plotting on top the morphological characters and see how it evolved according to the underlying DNA framework. And so, for instance, here we see here this red character is predominantly upper groups. You see the green one has evolved twice, and ancestrally we have that orange character state. And that can be interesting for various evolutionary scenarios. With biogeography, for instance, particularly with islands, with Hawaii is a famous example. So there we can map here the tree onto the different Hawaiian islands. And we can see that the shallower nodes are on the more younger islands. So we can really see an evolutionary progression in that particular group. With molecular clocks, we can eventually also define the ages of groups from particular organism groups. What is frequently the case is that the error bar is fairly large, but people are working on that and constantly refining those approaches. So then, in summary, so what we have in systematics, we have nomenclature, the naming business, taxonomy, how to organize these names into a classification scheme, phylogenetics, how to make trees. The valid species, that's probably what we're most concerned with, what shall we name that species? That is by community consensus. There's no commission that is agreeing that the correct names need only to follow the rules of the International Code of Botanical Nomenclature. And then these phylogenetic trees can be composed of all sorts of data, morphology, molecules. We use evolutionary methods here, cladistics based on the maximum likelihood. Then also remember again, we can rotate at the nodes, we can collapse these nodes, and polytones indicate uncertainty. Last but not least, once we have the tree and we try to make a classification out of it, we only use clades, monophyletic groups, so the ancestor and all its descendant, and should be as inclusive as possible. All right, whirlwind tour. We already dealt with a few questions, maybe there are a few more. So, yes, sir.
I'm super curious, there's certainly a lot of orchids that have some history of hybrid origin or the degeneration species in the first place. How do those end up impacting those models? And clearly, like you can find things that have very clearly different genetic histories based on chloroplast data versus ITS as an example. Like it's telling you the story that these came from different origins. How do those end up in those mathematical models of storing up your trees, so to speak, or contributing to dramatic uncertainty of where things should be placed?
Okay, once more for Zoom people, question of hybrid species, species of hybrid origin, how do they affect phylogenetic reconstruction? This isn't a problem, no questions. And they're basically in plants, that is much more common. In animals, basically ignore it. Maybe it occurs once in a while, but basically you can ignore it. In plants, it's a real issue, and that is basically also why you do these analysis with just the mitochondrial genes, with just the chloroplast genes, maybe with some of the nuclear genes, and then you compare the tree and see whether there are certain patterns where agree or disagree with one another. And that may then be a clue whether there might have been a species of hybrid origin. Now the determination, okay, let's say exactly this is the species that was of hybrid origin, that is a really tricky one. I think frequently the easiest way out is you just ignore the problem. No. This does not happen. So you just hope for the best, unless you can't escape that conclusion. You assume it's not of hybrid origin. How true that is, excellent question, but therefore essentially we need to have better analytical techniques that take into account what is referred to as reticulate evolution. So reticulate means you have a branching coming back together and making a reed, enclosed rhomb in the evolutionary history diagram. And that is really tricky from an analytical perspective. I have not seen a good way of really figuring that out.
So,
I'm sorry.
you have a question related to that? Then I'm gonna go a different direction.
Is your question related to the last one, or is he gonna do some that?
Yeah, it is. If you have two very similar species, maybe they're fairly different regions, and you're trying to argue that they're the same or that they're different, but the whole genome sequence for 20 specimens in one region and 20 in the other, so you got the very unique. How similar or different do they need to be in order to use the DNA data as a number that you can say they're distinctly different species or they're not?
Another excellent question. So my approach would be take a third species that's clearly distinct. And then you see, okay, what's the difference between the two that are clearly distinct? And how different are the ones that you wonder about? If you say the same differentiation between the two that is clearly distinct and the one you wonder about it, okay, then there's 3 different units. If the one that are sort of kind of a little bit different have a very shallow node, as opposed to one that's very clearly distinct, has a long branch, then that suggests to me they're more the same grouping. So that these 2 putative species are actually specimens of the same species. Is that 100% guaranteed? No, absolutely not. And that is why basically systematists will stay in business for a long time, because we will argue about that for a long time. Derek.
So I'm thinking about Oberonia, or actually selfishly, I'm thinking about zootrophians. Okay, so within like that specific genus, is there a number of informative genes that you think is like a magic number? Like within, is it like 2, 3, or are we talking you're using 500 genes and like 20 genes? There's at least in fungi, the more genes you put in, the more chaos, the more noise you get, and basically no resolution. So there's a happy piece. And what is that for? What's that? in your.
Okay, for Baroni, so we've done a little bit of molecular work, but little, with emphasis on little. And there, the biggest problem is material. And so unless you have material, you can't sequence paper, right? So that is one of the biggest problems I'm having with my particular group. In terms of what is the magic number, there's kind of two things that we should be aware is the number of genes and the utility thereof. Some genes are good for a certain kind of question set. And some other genes are better for a different type of question set, and that typically refers to of how old the splits are that we're trying to resolve. And so it's not necessarily the number of genes, but you get good data that have not undergone too much evolution, which is then referred to as saturation. And so in general, I think in the past, 10,000 base pairs is fairly decent, guesstimated.
Yeah, sure.
So
I mean, it
that's
depends. Is the reason that the inclusion of more genes causes more error or less precision due to the hybridization and the genes switching around in fungi, the swapping genes?
Okay, so this is like very recent stuff, but from what I can understand, it's not really well known. So when you take basically the fungal phylogeny as we know it today, and you add in hidden genes, and you can see, you know, with these housekeeping, well-conserved genes, you get this beautiful phylogeny that makes sense. But when you start throwing in other genes, it's almost like they each create models of their own. And that's what I'm assuming.
Yeah.
It's just noise essentially that you introduce, and then basically your signal to noise ratio goes down. And so, yeah, I don't think there is a magic number for that. And there again, this is basically the issue. Then also, once we have all data, like the whole genomes, what then? Then we need to think about data filtering to actually figure out what is good anymore. So is it actually beneficial to use all the data, or should we be selective? And that will be a fertile avenue for the bioinformatics field, of which I'm not part of. I also want to emphasize that. So this is Chris Thacker, my wife. She is doing a lot of phylogenomics with fishes, so she knows more about all the mathematical modeling and that sort of approach.
A lot of doctoral theses in the future.
Yes, indeed. So yeah, what I'm trying to do here, you know, I have 1 hour to basically get you grad school. So basically, in order to really understand that, that will take about 5-6 years typically. Well,
Yeah, so one thing I hope that you would go into was the difference between subspecies, varieties, and form, but that's.
sure.
But real quickly, natural hybridization has been found to be actually a major pathway of speciation. Mary and I did a book on the genus Californis. There's one that I don't even have 3 species involved. There's a common hybrid swarm, which is they keep going back and forth to either parent, and a third species come in that's only slightly related. It's the same subgenus, but it's so there's things like that. And so and then I just recently heard about with corals, the genus Acropora, the elkhorns and staghorns, that they keep finding all these hybrids, but really it's now they just refer to them as mega species. Because you can't name each different one that they find because it's just the crossing back and forth of genes.
And then it almost goes back to the question of variability, interspecific variability versus biodiversity. So are all these species that we thinking are hybrids, are they actually independent species, or are we getting something that's referred to as introgression, kind of the back crossing of insufficiently separated species? So yeah,
Oh my goodness, like, oh no.
So both of the mollusks and cowrie is one of my,
Right.
and so I have numerous natural hybrids of cowrie. I have a lot of them, so.
And then we get into the species concept, of course. For instance, hybrids, how much hybridization do you permit? And then the question about hybrid fitness, if one hybrid fitness, hybrid vigor, Or do we have reduced fitness? Then we get to the biological species concept, right? And abalone, of course, also famous for hybrids. Even some of the type specimens are hybrids, which is beautiful, of course, again, from a systematics perspective. Yeah, so unfortunately, I can't solve all of biological systematics in 1 hour. So, yes, it's a fertile ground. I hope I provided you with a little bit of a toolkit to actually get into it if you're interested in it. It's fascinating. I'm finding it fun and I'm playing around with it. And you can make a career out of it. So have fun with it. Right.
Let's see. Okay, do you want to do the online first? Yeah, okay. All right, so thank you very much. I think there's some real fodder here for us to do some fun stuff, fun offshoots, systematics.
Wait, I have to recover from this first. Straighten it all out.
All right. So what do you want to do first? Do you want to, where's from?
You might as well do that.
Do you want to do the online stuff first? And then we can move to the.
Who talked, did the people online, they talk about their plans?
Yes.
No.
I can do it.
Yeah, I want.
Yeah, you talk about it.
So what's really cool about this is actually a natural hybrid, a perfect example of a natural hybrid. Leilani Ansett by Leilani Albert, which is not an Albert on the table, so that's just a beautiful Pinkinian, and I believe it's supposed to be a wild one, a wild plant, but I've also heard that some of them are actually a man-made hybrid mixing up with what used to be Cattleya Burgundiana. So whatever, I think this one is supposed to be the wild plant one, brought by Dave Vermeer. We bring to the Lily will be end again. We're going to see the ants up here in minutes. These are the true leathers, supposedly what they're saying. Brought by Dave also. So the Lily Cattleya Black Forest, the individual, this clone is called Dark Splendor. So LC stands for Lily Cattleya. Depending on the parentage, they can all be Cattleyas now. So
Yeah.
but anyway, this is Cattleya Pumala, quite obviously. So. Heidi, who is, I mean, she was out in the other room. So this is Epidendrofolia folia, which means leathery leaves. And we have one, I think I saw one. So there's one in the table somewhere up here you'll see closer. So that's quite a common one. Jeff Harris brings in Miltonia, juicy fruit. So it's a pure Miltonia, I take it to be. But no idea, but it sounds like a natural hybrid. So when you see a name, it sounds like a natural, sounds like a specific name using the hybrid. It's either a very, very old hybrid when they used to be able to do that. Now they don't allow them anymore, or it's a natural hybrid by another one called Mato Grosso.
So Benodi is Miltonia.
Benodi, yeah, that's I don't know the hybrids very well. Actually, don't know them almost at all. For Paris, this is a really polymorphic. What I mean by polymorphic, maybe not the right terminology. Each species is extremely variable within this genus. So all restrictors are really confusing. And mostly it's the labellum and the lip is what differentiates that, and also of course where they're from. So this is the Gudulata, the Shirky Gudulata, and the one in the background is Nidiorinca, the little yellow one up there at the top. You can see that one almost never really opens up fully. Actually, that's a fully open flower there. So you can see the size difference in the genus, size difference in flowers. Judy Drew brings in this Mastoid decumana. I believe it's a Peruvian species, a beautiful thing. It was quite rare initially when it came out, but now it's easy to grow. It's one that, it's an easy one to share. It's a beautiful thing. And the thing I like about orchids, you can pick and choose what you want to grow. And I love plants with flowers bigger than the plant they come from. I mean, that's great. So here's the Judy brings in a Mastoid strobeli, which is an Ecuadorian species named from the strobel. So real quickly on a scientific name, a name like that, strobeli, the small s thing. It's named after a man With us in Strobel, it's I or II. Same if they're ladies, AE or IA, so there's ways to tell. And if it's IANA, I think it's just named in honor of them, and I think there's other, I think there's Orm too, anyway. That's another way to tell about what names do. That's a beautiful thing, it's fragrant, it's easy to grow, but a plant like that can bloom itself to death, literally, they can have so many flowers, you go, oh no, let it stay on too long, the plant's gone, it's dead, so it'll do that too. But it's beautiful.
I want that.
I say golden monarch, it's I think of Viciana and McClure, I think. McClure is the largest flower of all of the masturbates of all 550 plus species. And golden monarch, I believe it's got, I don't really know, but I think it's got McClure, I think I want to say Viciana in it too. So Sharon Omasaro brings in,
Trader Joe's, Trader Joe's.
Trader Joe, I got that. I wouldn't eventually, okay? Oops, there, yeah.
Trader John.
Trader Joe is-
I, right? Because he's a male.
Ensis denotes where it's from, Ensis means it's from Trader Joe's, yeah. I did it really right, they
Okay,
actually
now you're.
researched that, yeah.
You got it right.
That's a very nice.
So Peter Meyer brings in a lily, Ensis Guerrero, so this is actually got a, I think it's, I want to say it's subspecies Dawsonia, I believe, if I got that right, but anyway, it's a regional population. It's actually warmer growing, it's not as cold tolerant as these, we both seem to do okay with them in our area. I'd like to be a little bit warmer, but it's known as like kind of the splash petal or the flame tipped variety. Dave brings in this Cattleya persevilliana, this is the clone summit. This is the Venezuelan species, it's one of the more cool tolerant of them. I think this can go easily down to 50 or below quite easily, especially if it's kept dry, so. Has a weird smell, it's really unpleasant. It smells like kind of like wet clay or something, it's so odd, but not unpleasant. Dave brings in Cymbidium docido, okay, anyway, don't know much about it, the red Cymbidium hybrid. This is great, oh my God, Stuart's on this, it's something on my list, this is a Chilean species. It's a flora, it's a genus of probably almost 100 species, it's Argentinian, mostly Chilean. I think some even come up further north, maybe into Bolivia, I'm not positive of that, but this one is Magellanica, and those flowers are not small, they're almost, I think like 2.5 inches. They're quite stately, and I just think that's outrageous looking, so.
Yeah.
My want these days, I've been growing orchids, this is my 50th year of orchids this year. And my want now is to go see them in the wild, I want to see things I won't get to see on the table. That's one of them. Another one, orange, not as big flower, but really cute. And Gavilia, so Gavilia, there's another genus called Bipinula. So the three look like they might even be in the same genus eventually. Anyway, Gavilia is another one of the Chilean, Argentinian genera of that group. Gavilia lutea, Stuart, man, he's on a great set. So Gavilia lutea.
These look like they're all in the wild.
Yeah, they are.
His greenhouse.
Good things actually, Gary.
Really big.
It's got a great backyard.
Yeah. Really good job. Golden orchid is actually the furthest, I think it's the southernmost occurring orchid in the world. There's a tie with that with some little helmet orchids that are from these islands that are sub-Antarctic islands. So this is one of the most furthest south of all orchid going furthest. How do I say it? Southernmost orchid, one of the southernmost. Golden orchids list on the end.
What group is it in?
I think it's related. I want to say it's related to the Gloria, but the Gloria, that whole group is related to the Australian. The tribe is the Donkeyorthaeridae, and it's a sub-tribe of that, I think. If I got that right. I'm not sure. Did I get that right, Daniel? You know? Okay, all right.
So you're saying something off the tip of South America is related to things in
Yeah,
Australia?
I think
Like
yeah.
Well, that's really interesting.
There's a lot of families.
I think of like Southern Africa.
Right, so the Protea family is only in the southern hemisphere.
Right.
So it's in South America, South Africa, and Australia.
All right, that's all. That's all.
Yeah.
I like it. I think it's great.
Yeah, don't break up that group.
Zoom in with phone.
Yeah, I know.
But I, you know, my parents. Ladies.
First, I gotta find you.
And share a picture of that.
He's rearranging her stuff here, so.
What? Yeah, just not.
The polish do. You know, what we used to do, this like 3 years ago, I kind of missed COVID completely. I didn't miss COVID, I just didn't come. So we used to raise our hand, so Paula brings in this dendrobium, one of the green hybrid. It's also, looks like it's got, sure, looks like it's got nobly, and it seems like some nobly crosses in the same section of dendrobium. Very nice hybrid as well, they're cool tolerant, so.
Smells great, and it's been in bloom for a month and a half.
Good stuff. Brett Anderson brings in this Fragmipedium species. Fragmipedium is a new world. So the lacehoppers are their own subfamily. There's 5 subfamilies of orchids. Lacehoppers are one of them alone. There's only 5 genera of lacehoppers in South American, or you consider the South American tropical, Central and South American tropical cases. It's Fragmipedium walsay. Fred, everyone knows Fred.
And Ron, just because we were talking about all that confusion with naming, remember when Wallacea, Lindeniae, and Marthusianum got all confused?
Right. I think it's still up there, right? A little bit. So Paula, she brings in this Cymbidium. Yeah, oh, the name. So Devoniana is a really nice little species, really weird, not related very much. They distinctly finished like this is a Devoniana hybrid, so no name other than that. Involving Devoniana, not a miniature, but a very small, quite small Cymbidium species.
Then I thought.
Well, I'll bring in this Botanara or Socoletta Cat Cosmic Delight. So, no, I guess it's a code jewel by Cosmic Delight. So.
Can we just move the owl?
And Anderson brings a species that you used to see more frequently. This is Iria javanica. So Iria is a genus that got split in a bunch of genera and they stuck. They worked out. So this is quite a variable species. I was showing Jan some pictures and I forgot. I had it's almost red one. One is pink with darker stripes. Really quite interesting. Heidi Arna brings in this, we saw this in the show and tell, someone had it in the show and tell, this is the epidendrum folium. You see the leaves are built, they're not as leathery as some things, but that's its name.
It smells like parsley.
Oh, is that what it's
Hang on. Let me rename it, because you're behind it. You have to be very.
I think you gotta get a little.
Where am I?
Here, why don't you pick a spot and I'll move it to you. You pick a spot and I'll make that.
Is Heidi in the room? Yeah.
You pick a spot and make it go there.
Alan, yes, where's Alan? I did, sorry. It's been a while since they've been here. So Alan brings in this Paphiopedilum species, Patrixianum, comes from China, and I think into Vietnam, is that correct?
Pick a spot.
And anyway, it's interesting, is I don't know what they're concerned about the one really widely, that's still considered Patrixianum?
Yeah, we were just talking about it. Apparently that still is in
I think
cultivation.
they were thinking it was like happening for a while, I don't know where it ended up. So that's Alan's plant. I brought in the Mastodontine called Angulifera. It's a slumming species, and it's this is I didn't get the award, but it's the award JNL. The clone is JNL has an AM.
Doesn't have any flowers though.
So as like, and sometimes it has like this banana oil fragrance to it. I like that. It's quite a good. I brought in those plants. I know, right? So it comes in a bunch of forms. There's like a yellow form. There's a bicolor form with the tips of the. It's kind of triangular. There's ones with lighter colored tips. It doesn't matter. So. And it brings in a Oncidium chlorophorus. It's kind of petering out, but this is one of my very first orchids, and everyone goes, oh, that smells like lemon pledge. No, it smells like stale beer. So it's a stale beer, lemon pledge kind of fragrance, and it's really nice. It's made from hybrid twinkle, I think.
Yeah.
What I remember. So that's pretty cool. And what do you think it smells like?
I would say maybe a little bit like stale beer, a little bit like lemony though.
Yeah, exactly.
Yeah. This one to me stinks, but my husband can't smell it.
This is a bulbophyllum. I don't know where that name came from, but that's actually a bulbophyllum polyflorum.
Oh.
Yeah, so it's one of the Ceropetalum types. What's that? Ceropetalum types, the ring of flowers.
Yeah, it's like, yeah, it's tasty.
Is it? Excuse me. Yeah, it's okay.
It's, but I accidentally, so I had a grow light fall in my little indoor greenhouse and it burned off all the leaves, but then it still bloomed.
I hate cilantro and I have this dendrocolum. It smells just like cilantro. It's like, I love the flower, but I hate the smell. It's like. All right, we got a half here.
Yeah.
Let's go for this
You
one.
can put it out.
Yeah.
Yeah.
Drop it.
Right.
No, that's not. The leaves are inside, that's great. So there's one of the things for here.
Yeah, that's somewhere.
Here's the tag, Ron.
Lower.
So this is a, what's called, it's not quite a primary hybrid, it's a cross between two species. And one of the parents of the species and one is a complex hybrid called Madeline Giselle Lee, it looks like. Anyway, so that's, the spice is really obvious because the shape of the dorsal, it pinches her right here. And, just the shape, the overall shape of it looks a lot like spice random, with different colors and the bigger flower.
There, he's gonna be nice there. Okay. There's, opportunity.
Okay.
It's there.
So why is it a hybrid though? Did they tell you what it was?
And it's like process itself.
So that's, so when it's a smelting,
Yeah.
it's a sibling cross, so two of the same species per cross to make a sibling cross. A selfie is when you just take the pollen, put it right back
Which one
on
do
the
you
same flower.
You're the choir.
What is things?
Apple honey, apple honey. White spread species all throughout, in Thailand, but it's also in some of the, Indonesian islands and such.
I think it's also synonym with Hainan, right? Like
Yeah, yeah.
Hainan, the island.
Oh, good. Oh, it's this guy has a strap. Anyway, this is a Honda, but I don't know what actually it is. Obviously.
That's the Indian island.
Yeah, that's exactly, but that's really.
All right. Okay, so this is a really cool species. This is a little plant for you luminary. This is called Phycocentron gokusingii. There's Marillianum and Bossii now that there. I mean, there's really more, but all three of those have the same kind of really smooth fruit flower. Nice.
I actually didn't even notice the flowers
Yeah.
for a couple days,
Right.
but they were on the bottom and I was watering and I was like, oh, wait, there's actually flowers.
Plants actually plant
Yeah.
2 feet tall at least, and then when they get like that, they start puffing quite prolifically. So Bill Weaver brings in a lily ancestor. This is a form called lineata. So it's got these streaks on the edges of the petals. So real quickly, so Laelia.
It's a little tiny Laelia.
Yes, it's a little tiny
Yes.
Bill Weaver miniature plant.
It's almost too big.
So real quickly, somebody mentioned Laelia. So supposedly, the Rapingus Laelias, the Puma group, the Cattleya, well now Cattleya purpurata group, and Cattleya, formerly Microlaelia lungii, those are all now Cattleyas, and then Laelias consist of the 6 or 7 species, maybe more, of the Mexican lilies. That some rubescence goes down to Central America, down to Panama, I believe. And it also now includes what we used to know as Schomburgkias. So Schomburgkias have lily-like bulbs, whereas Myrmecophilus have the cowhorn bulbs that are hollow, have a stinging, they're home for stinging leaves. So that's a whole separate genus. But that's what happened to. There's no Schomburgkia anymore. So now what used to be Schomburgkias are now lily. So I'm gonna go back there, but that's their good like stays back there. We have another Mexican. Oh, wow,
Look at the petals on that thing. I think they're wonderful.
So this isn't the pink Indiana, part of my Paula, right? No?
It's beautiful.
Okay, well, it says it's Pink Indiana, which is that thing we saw first of the online thing. I've never seen anything like it.
I've never seen anything like it either. I love
But
it.
Anseps has four-sided pseudobulb. That's one thing you tell about an Anseps plant. I can't tell if it's exactly four-sided or not, but it always has four-sided pseudobulbs. So it looks like an Anseps with a little something else in it, but it's still really pretty though.
I love the petals on that. I've never seen
Yeah,
that.
that's
on
very cool.
anything.
We all want it.
I want a division. A trade.
Okay, so you're showing this one?
It's showing the pot right now.
Sorry, I'm walking weird. I got a pinch in my neck and it's affecting my left arm. It's going, it's tingling. So Bill brings in a Australian dendrobium hybrid, purple zip, SVO stands for Sunset Valley Orchids. I just was there last week. It was very cool. The German King Zip, red splash. That one parent has an award of merit from the Orchid Society, AOS. So anyway, very cool. Blooming early, which is pretty amazing.
It's the only.
That's weird,
King
but I see a
Anna
lot of
blooming
things. My King Annas are spiked. Right now.
Yeah, I got them in spike. That's the only one that's fully open.
They don't even know what's going on. So Bill brings in a surprisingly complex hybrid. So I thought initially it was a Cattleya milleri. It's a Brazilian tangerine by Siegel's SVO. So another Fred Clark hybrid. He likes a lot of the mini cats.
One assumes it's Millerai in it because of the color.
Yeah.
And the
And
bulb
the
shape.
bulbs, everything about it, yeah. Vince Pietro. Now you said your name. Oh,
There you go. Get the one that's got the lip. There you go.
If you aren't an orchid, I'm sorry, you're out of luck. So this is the Brassolalia Cattleya. So that has a Brassivola in it, so a true Brassivola, Martin Dior Halcyon. And that's times the Brassolalia Cattleya, guess what, is that, that's heritage? Okay, two are without lips, is that genetic or something has an
It's
accident?
the first time I've had it for like 5 years and it's always had fully lit flowers.
Uh-huh.
So this year it threw out a little.
Do you have a cat or anything?
No, I was watching it and I was like, wait, where are the lips?
All right.
I have a question about Brassivola. I just bought something and they have a Brassivola glauca, isn't it Brassivola glauca?
No, Benrica Lily Glauca, it's the only two species, Digbiana and Glauca, the only two, and it's been like for 20+ years, yeah.
Yeah.
So they're pretty distinct looking now when you look at the plant.
Mine still says Brassivola on the tag.
Yeah, you would.
Yeah.
So Kay brings in two species. She brings a Prostechia vitulina, which is one of my favorite Mexican Prostechias. I like it because of the color. Interesting, Mexico has a opposite regime of our weather. We have a Mediterranean climate, so cool, wet winters and long, hot, dry summers. Mexico has the opposite. They have wet summers and cool, dry winters. Yeah, this one's from some area of Mexico. I think, did you see it, Mexico, Mary? It's from an area where it gets moisture almost year round and it's really high growing. You grow this outside in the berries, does really well. I've got two different ones. One of mine's actually reached the point where now the spike gets branched, but it's a cool growing orchid. It doesn't like a lot of heat.
What's it called again?
Prostechia vitulina.
That's the Chiapas.
It could be. I don't know what state it's from. And then Kay brings in this Prostechia pygmea, which it really is.
That's pygmy, all right.
I don't know if anyone's made any hybrids of it.
No, it changed its name recently.
To what?
It changed its name recently.
To what?
Last time it bloomed it with chlamydia.
Armidia pygmea? Yeah, that's an old genus name. Yeah.
You need your sevronoscope to make a hybrid out of that.
So Kay brings in this too. This is now an Isabelia.
It is?
Yeah, it has been fought Violacea. So interestingly, the three species in that genus look nothing alike, and yet they found natural hybrids of this one by Virgin House, the one I think it's one with the little netted pseudobulbs, the little onion-like plant with net cross net pseudobulbs. Yeah, that Violacea is Wisong. It has a something maybe Anna Chai's awarded HCC.
Yeah, I got that.
Yeah, it makes sense. Yeah. He also brings in Dendrocolum parvulum and Ornithocephalus estrada. So so interesting with the dendrocolum, people like they're in Australia, they love dendrocolums and they hate that they went into slide. And that's like, I hate it. No way, I'm never going to do it. But interestingly, the pathway, evidently it's 3 different lineages, dendrocolums, 3 different lineages. It's like one cohesive group, it's 3 different So that's why they put it in Salagi, whether it'll stay, whether that's enough, who knows, but they're not, it's not a monophyletic, what we're, it's not a monophyletic group as that genus, so Dendrocolum is no longer. I agree that something has to be done with it, because there's 3 different lineage is,
Yeah, that one's from the Philippines.
yeah, it's one of the smallest of the Dendrocolums. And Paula, your plants are all over the place. So she reads the medium.
Ensign knuckle.
Ensign knuckle, Paulista, is that you?
No.
And as a B from the CSA with a B.
Bronze.
A bronze, so it's a show trophy, or I don't know what they got.
No, it's a,
It's,
it's a.
it's an HCC for
For Cymbidium.
Cymbidium.
Okay, thank you. This guy,
Because you had Tracy and.
so,
Probably.
okay, if I get this wrong, if I mess it up, Nancy Garcia von Kriegenberg. Okay, cool, so this is a Dendrobium mini snowflake, so what's cool about this, it's a Laturia, so Laturia is a section with a huge genus, it's the section Laturia. There's about 45 or 50 species, they're almost all New Guinean or that region, and they range from the mangroves up to where it's quite cold. And they always have a bulb that's got a few leaves on top, and a bulb that swollen and tapers real narrowly at the bottom. The hybrids last, the species and the hybrids both last about 3 months when they bloom, so they're really good plants. And what's nice about them is they all stay really close together. They don't like send runners out or long rhizomes. They make nice clustered plants. So they last a long time. And Nancy also brings in a no name, I call it leopard orchid. Yeah, I call it that too.
Yeah.
I'm sitting, well, I told you if you're on sitting now, I think it's Pulmonara, right?
It's
I
like
don't know what that is.
wildcat type.
No, I think it's a Pulmonara. Justin, hi Justin, how are you?
Yeah.
He brings in, good, he brings in some, he belongs to the Prothollus, a lot of us go to the Prothollus Society meetings every few months. And, totally good with that, 2 months. And it's a big party basically, so he's been coming, which is great. He brings in, Mastoid brachyura, which actually means short tails, they have little short tails, a little miniature, very, very cute. Best grown on the mount, because a lot of the times the spikes hang down.
Sorry, good looking, the next one on camera.
Why don't you come over here, Ron, stand over
All
there.
right, it's been a long time, okay.
Then-
It was never in this room before, it was always in the other
Then he
room.
can zoom in on them while you describe.
All right, sounds good. So Justin brings in this, platystellic oxyglossate, nothing amazing about that, it's tiny little flowers, oxyglossate means pointed tongue.
We always use the table. There you go. I thought it was a mosquito.
Behave yourself, okay, just because you grow giant plants.
Specimen is relative.
There you go. Sharon brings in this, Phallus sequestris Montclair pyloric, which is interesting because it's a natural, I know it's not natural, probably, lab produced, not made to be pyloric, but it's probably a mutation that happened during a cloning or anyway, so it's a species from Philippines and Taiwan. It's very variable. There's a regular form and some red and rosé, where it's all pink with a little bit of a yellow palette. So quite a variable species. The cerulean, albas, semi-albas, pyloric. Really nice species though. Sharon brings in the Mastoid floribunda from Mary Garrettson. Mary's back there. Sharon's got this plant from Mary. I think Mary has her on here too.
Who's a beat?
Flash.
So Sharon brings in this Mastoid striatella. It's interesting because when I first started doing Mastoid, it was called E. caudata, which means without tail. They're little short, stubby little things. Nice, really nice species, kind of variable in the size of the flowers. And it, sometimes it smells good, and sometimes I don't think it has a fragrance, so.
That's good.
And Mary's plants, we got a Stelis argentata. I had to laugh, someone asked, is that Stelis argentata?
This is Sharon.
Okay. But yeah, we did that, that's the one that we did. There, yeah. So someone said, Mary, is that Argentana shit? Yeah, I don't know what Argentana is. It's like, that's what they call every Stelis almost. Argentana just means silvery. I don't know, I guess maybe the vaginal flowers. I don't know what.
It's the edge on it, see, look at the flower, there's.
You know how many Stelis do that?
Probably a lot of them.
All of them.
A lot. Could be though, I'm not saying it's not, it's at the left, but everybody calls their Stelis Argentana, even though they get 800 species of cheese flowers. So that's Mary brings in Mastitis mystica. I love this group of Mastitis. It's called the Saltatus group. It's characterized by this little pot belly. And it's supposed to be a natural hybrid. I don't know what it would be made from because the color is unique to this group. So I've not heard you called X mystica for a long time. She brings a whole slew of Mastitis and other pluralities, I think. But she brings in the Stelis Cypripedioides, which means like a Cypripedia because of the pouch-like lip on it, pouch-like sin sequel, it's not the lip, everyone thinks it's the lip. Cypripedium, Latest virginis has a pouch-like lip. This is a pouch-like sin sequel. A lot of profiles have this, both flower sequels joined together, what's called sin sequel. Mastoid Garcia is really cute. It blooms quite heavily. It's got really cute flowers. And they always seem to be in pairs, occasionally 3, and they have a triangle stem, which means some of the spikes will live from year to year and bloom more than once. And this is this is something I just kind of discovered. Mastitis cetacea comes in a purple form, and I thought these white ones for a long time worked. I think this is actually one called Bacillifera that I verified just recently. It's one that a lot of people have mistaken as cetacea in their collection, but it's related to caudata, which is a very well-known species. This is also in the caudata group, Mastitis nivea, which means like cloud white, I think. Very, very pretty. Actually, Mary, I think this might be this is actually Xanthina pallid that has a real distinct black pepper fragrance. It has a black pepper fragrance.
Okay.
There's nothing like snow, actually, maybe
Yeah, it's nivea snow, but that's not nivea. I had to look at it again. They're very close. This is one called Richardsoniana that Mary got from me. It's a little Peruvian species with triangle stems. This is one called Asterotrica, which means star-like hair. So evidently the bottoms. Look up close and yeah, this hair is like just like little stars. That's what they call it. This is a little tiny. Can you get that one? But it's still the Argentosa, I believe, and it's I mean silver. You know what's silvery about it?
There is a flower in there.
There is. It's your favorite. This is your favorite one on the table, right?
Yeah.
Favorite, right?
Compost.
That's mean. You mean.
It's one of those cool growing ones. Cool growing ones. In my greenhouse, it's compost.
Mean. This is Mary's farm. I didn't want as many pieces she got of that. So that would be the one. I got this from a friend, and I gave this to Mary for a present recently. And this is Silly. Flora, which is great because these spikes last for a year. The next year spikes come up. It's still blooming from the year before, so that's pretty cool. And yeah, and this is another one. This is another Steli. This is a warm from. It's actually seen as from warm, intermediate, cool. It's called Stemastachia. It has keeps going on and on and on and on. And those are all those little side branches on the inflorescences. Those are all work with well formed flowers, and it just seems to grow and seems to bloom. Being constant of it, Stemastachia. And this is a Andinia. Andinia is a really strange genus because there's all different looking things. And those flowers are this species comes in yellow flowers and orange flowers, same species. And I haven't seen anything in between. And it's very cool growing and not easy to come by. Right at the table. Cool.
Thank you, Ron. Okay, for those of you at home, that's it. For those of you here, get your raffle tickets.
Time to go buy raffle tickets.
Okay, we're gonna take about 10 minutes break, buy your tickets. We're going to raffle.